Welcome to this model. In this model, let us have an insight of biopotential electrodes. Electrodes are employed to pick up the electrical signals of the body. In presenting a bioelectric event to a preamplifier of the signal processing unit, a pair of electrodes plays the role of a transducer. Since the electrodes are transferring bioelectric event to the input of the amplifier should be designed such that it accommodates the characteristics of electrodes. And this type of electrodes to be used depends on the anatomical location of the bioelectric event and the dimensions of the bioelectric generator. And the electrical characteristics of the electrodes specify the type of preamplifiers. When the microelectrodes are employed, then there is so many restrictions on the input impedance of the amplifier. Since they have large impedance, high resistance and low capacitance input circuits are needed to transfer the bioelectric event to the amplifying system. And bioelectric electrodes acquire the signals like ECG, EEG and EMG. Here are the three types of biopotential electrodes, known as microelectrodes and skin surface electrodes and needle electrodes. Electrodes used to measure bioelectric potential near or within a single cell are called the microelectric potential. And this microelectrodes, their tips is sufficiently small to penetrate a single cell in order to obtain the readings from within a cell. The tip must be small enough to permit penetration without damaging the cell. So the microelectrodes are of two types. One is metal microelectrode or micropipette. Metal microelectrodes are formed by electrolytically etching the tip of the fine tungsten or stainless steel wire to the desired size. Then the wire is coated with an insulating material. And the metal or microelectrodes, the measurement requires two electrodes. Resulting voltage potential is the difference between the potential of microelectrode and reference electrode. And the total sum of the three potential E is equal to EA plus EB plus EC. And this micropipette, it is a glass micropipette with the tip drawn out to the desired size. Micropipette is filled with the electrolytic compatible with the cellular fluids. So this type of microelectrode has a dual interface. One interface consists of a metal wire in contact with the electrolyte solutions inside the micropipette, while the other interface is in between the electrolyte inside the pipette and fluid inside immediately outside the cell. Okay, and this electrode, a thin film of precious metal is bonded to the outside of the glass microelectrode, and the metal electrode interface is between the metal film and the electrolyte of the cell. Here the potential voltage is given as E is equal to EA plus EB plus EC plus ED. And next let us have a look into needle electrode and depth electrode. Needle electrodes are used to record the peripheral nerve action potential, and they generally simply inserted through a small section of the skin just beneath the surface and parallel to it. Okay. Depth electrode studies the electrical activity of neurons on the surface of the brain, and this type of electrode consists of a bundle of Teflon insulated platinum and iridium alum wires. So, and for AC insertion of electrodes into the brain, the end of the supporting wire is round shaped, and the number of individual electrodes forms the electrode array or bundle. In this bundle of electrodes, the end of each individual wire has the individual electrodes. Applications when it is used to inject medicines into the brain and to measure the oxygen tension. Figure shows some of the examples of needle electrodes. And as I said earlier, needle electrodes records the peripheral nerve action potential. It resembles a medical syringe. So at one end, a short insulated wire is bent, and the bent portion passes through the lumen of the needle. The setup goes into the muscle. Now the needle will be withdrawn and the bent wire remains inside the muscle. Needle electrodes may be of three types: monopolar electrode, bipolar, two types. And but this bipolar electrode, it has one reference electrode and one active electrode. And the main applications of needle electrode are it is used mainly in EEG and EMG signals. And the next category is surface electrodes. Surface electrodes measures the potential available from the surface of the skin. And there are 5 types of surface electrodes, as you can see in this slide: metal plate electrode, suction cup, adhesive tape, and multi-point and floating electrodes. The diagram below shows the metal plate electrode. This Metal plate electrode are rectangular or circularly shaped, but it has a small contact area and do not sit completely on the patient. And the main disadvantage here is the slippage or it is very sensitive and leading to measurement errors. So nowadays these are not used. And the next one is suction cup electrodes. They are used to measure the ECG from various positions on the chest, and they have a good contact surface and high contact impedance. And due to infection or cleaning procedures, these electrodes are not now used. This is the diagram of the suction cup electrode. And the next type is adhesive tape electrodes, and the pressure on the surface against the skin squeezes out the electrode paste. Okay, and to avoid this problem, we use adhesive electrodes, and it is a lightweight metallic screen, and they have a pad behind the electrode paste, and this adhesive backing holds the electrode on place and tight, and it also helps to avoid evaporation of electrolyte present in the electrode paste. So the diagram shows the adhesive tape electrode. And a multi-point electrode are very practical electrode setup for ECG measurement. It has more than 1000 active contact points, and it has a low resistance contact with the human. And in any environmental conditions, the doctors can use multi-point electrode. And the last category is floating electrode. And in metal plate or rim electrodes, the major disadvantage is the movement of errors. Motion artifacts occur due to the motion at interface between electrode and electrolyte, and the interface gets stabilized using the floating electrodes. So floating electrodes do not contact the human subject directly. They contact the subject via electrolyte paste. So the main advantage is mechanical stability. Thank you.